Welcome back. In this video lecture, we will look at basics of classical and emerging tools. Okay, we will look at. We'll try to give you some insight into some of the classical and emerging tools. So why we should understand these tools now in the module 1 itself, right? So that is the question that one may ask. So the idea is we want to introduce this the give overview of these tools now itself, so that as we move on, so you will be coming across, you will be dealing with many material systems. Let's say you will come across, let's say you may work with, let's say, colloidal particles, right? Or you may work with surfactant systems or polymer molecules, polymer systems, right? So you may be wondering, what kind of techniques, what is the right technique that one should use, to characterize them. Let's say somebody may be interested in, understanding or performing the size distribution analysis of given particle system, or somebody may be interested in knowing what is the morphology of the given nanomaterials. Okay, some may want to just understand the structure. Okay, so depending on the application and end use, one would want to use the right technique and to characterize the materials, different nanomaterials, or the materials that you will be dealing with. You may want to know what suitable techniques one has to use, right? So this is the right time for us to introduce some of these techniques. Are very relevant to interfacial engineering now itself, so that as you move on, you will have some clarity about the various tools that are available. Okay, yeah, let's begin. Yeah, so first let us start with a brief overview of length scale. Okay, as you can see, our human eye, we can uh, you know visualize objects, right? Any objects, up to certain length scale only, through our naked eye. Say for example, anything beyond, let's say, 200 micron or maybe 300 micron, it is very difficult to resolve through naked eye. So one should use, resort to the modern techniques that are available. Let's say you may use light microscopy. Okay, so that will be helpful to resolve any objects of size, let's say, up to 200 nanometer. Okay, so that with that you will be able to uh, you know uh, look at the samples. Let's say for example, you want to see bacteria, how bacteria look like. Okay, you can see using the light microscopy. But if you want to understand, let's say, virus, how virus will look like, then you may not see them through the light microscopy. So in such case, you may have to resort to the electron microscopy. Okay, so with the electron microscopy, you may be able to resolve up to let's say zero point 0.1 nanometer. Using the transmission electron microscopy, one can resolve any object, a specimen of size up to zero point 0.1 nanometer. With the FESM or SEM, one can resolve up to 1 nanometer, right? So these are various length scale that you can connect well with the different living matter also, right? And on the right hand, you can easily map what kind of technology one should use, and what are the for example system. Say for example, you can use radio frequency up to about 100 micron meter. So when we talk about 100 micron meter, you can relate with the our hair, right? Our human hair, hairy particle is in the size range up to 100 micrometer, right? It is it is more or less same as 100 micrometer or slightly above 100 micrometer. So it is easy for us to resolve, right? I mean, you can see up to 100 micrometer through your naked eye. We are able to see your hairs, right? Hairy particle, right? But beyond 100 micrometer, then one has to resort through different technology. Say for example, less than 100 micron meter, let's say up to 10 micron meter. So you can give an example of epithelial cells, right? They are slightly above 10 micron meter. So you cannot directly view them through naked eye. So then you need some technology like light microscopy that will be of use, right? So you can resolve them under light microscopy. Okay. And so the bacteria and red cells you can easily map. Say bacteria is about 1 micron in size, right? Whereas the red cells is slightly above 1 micron. Okay. So you can easily connect, right? Now between 1 micron to 100 nanometer, you can give some example systems, that is like mycoplasma. Mycoplasma is a good example. And then comes viruses. So between 100 nanometer to 10 nanometer, there are various viruses. Okay. Range between 10 nanometer to 100 nanometer. You cannot see them under light microscopy. So one has to use special or modern, sophisticated technique like electron microscopy, to look at them, to visualize them. Okay. And if you want to relate anything between less than 10 nanometer, then you can talk about proteins. Protein molecules are of this of the size range up to 10 nanometer, between 1 nanometer to 10 nanometer. If you're talking about 1 nanometer, you can connect easily with amino acids. Amino acids are of that range. Okay. What about less than 1 nanometer? You can talk about atoms, right? So if you want to look at the atoms, right, of size, let's say 0.1 nanometer, one can use electron microscopy, right? So these are various length scale and various example systems, okay, to relate with the length scale, right? Okay, let us now look at the optical microscopy. So, thanks to the discovery of this, the people who invented optical microscopy in 16th century itself, but the much development came, after 18th and 19th century, right? So still lot of development taking place in this in this domain, that is optical microscopy. So optical microscopy is very useful technique to visualize any object. Okay. You know which are not possible to look through naked eye, right? So, say for example, if you want to understand how the bacteria look like, you can easily look at them under microscopy. Okay, some of the particle system that you want to, let's say, which are let's say 1 micron in size or 2 micron in size, you can look at them easily under microscopy, optical microscopy. Okay, all you need is just one light source and magnifying power. Just like how we use the magnifying lens, you have objective lens in the microscopy that has got the magnifying power. And whenever you shine a light on the object, okay, whatever is transmitted from the object or specimen is collected by the objective lens, and that is what is reflected in the the viewing lens or eyepiece. Okay. You can also fit a camera, digital camera, and collect those image and project it directly on the screen, computer screen. You can also record, or you can also record video of the live cells, right? That is also possible. You can also click capture a snapshot of the samples, right, using this digital camera. Okay. So these are very much possible nowadays. and there are some advanced stuff also, but the basic of microscopy is you have got some important components like condenser. So what condenser does is it collects the light and it focuses the light onto the specimen, and there is something called specimen stage, and on top of which you will keep the sample, and there is objective. So objective lenses are available at a different magnifying power. Say for example, you have 5x objective lens, 10x, 50x, up to 100x objective lenses are possible. So when I say 100x, 50x, meaning you will be able to see specimen, you know, uh, several times. Say for example, when I'm viewing the specimen through 10x objective, and my eyepiece is also 10x, right? So then I would be able to see the object uh, 10 into, so 100 times more than the object, right? Say for example, if I look at them through 100x objective, I am magnifying the object specimen by 1000 times, right? So that is how you are able to see the specimen very clearly, okay, which is not possible through naked eye, right? So these are various components. What is more important is that there are a few, I mean, many types of microscopes that are available. Okay, let's say for example, the commonly used are inverted microscopy or upright microscopy. Upright microscopy is available in two category. One is transmitted mode and another one is reflected mode. Whereas the inverted microscopy is available mostly in transmitted mode, right? So whenever you deal with upright microscopy, you will see that the objectives are fitted at the top, okay, whereas the light source is coming from the bottom. In the case of inverted microscopy, you will see that the objectives are fitted at the bottom, whereas the light source is coming from the top. Okay, so that's the difference that you will see whenever you deal with upright or inverted microscopy. Mostly biologists use inverted microscopy. Biological scientists use inverted microscopy, whereas material science scientists prefer the upright microscopy. Sometime uh, the upright microscopy comes with both reflected as well as transmitted features, so that you can see a sample of any kind, transparent sample as well as opaque samples. Okay, so more importantly, you must understand what is known as the numerical aperture. So numerical aperture tells us about the magnifying or resolving capacity of the lenses. Let's say the more the numerical aperture, uh, you know uh, greater are the magnifying or resolving power of the lenses. Okay, so the maximum angle that can go up to, in this case the alpha can go up to 90 degree, which means the maximum at maximum numerical aperture case, the sin alpha will be 1. Let's say if you deal with oil immersion objective, this eta is refractive index, right? So this may vary between 1.3 to 1.4 for the oil immersion objective, right? So this is about numerical aperture. There is something called Abbe's diffraction limit. This sets the limiting point. Let's say if you want to resolve some object, okay, which are kept at a distance of 200 nanometer, you will be able to resolve them under the microscopy. Say for example, if I substitute lambda as 500 nanometer, because this light microscopy works in the visible region, visible light. So lambda can be 500 nanometer. Then Na, if I put 1.4 here, I will be getting about 178.6. So one will be able to resolve a specimen which are kept at a distance of 178.6 nanometer. Okay, that is the resolving power of this light microscopy. The limitation is coming from the wavelength of the light itself. Okay, right. So let us look at types of optical microscopy. Okay, so more importantly, you will see what is bright field, dark field, and fluorescent microscopy. Okay, so the difference between bright field and dark field is, in the case of the dark field, uh, the objective collects only the scattered light from the specimen, which means that uh, the light does not directly fall on the sample. Instead, the sample is illuminated from the side, so that only the scattered light comes out of the sample, is collected by the objective lens, right? So in doing so, the because all light that are transmitted from the sample are not collected, so the background here will look very dark, whereas the specimen will appear bright. Okay, this is because of this feature. In the case of bright field microscopy, why we are able to see the specimen distinctly is because of the differences in the light absorption. Okay, although both will appear bright in this case, because specimen absorbs light in a different way, whereas the background absorbs in a different way. Because of these differences in light absorption, we are able to see the specimen distinctly, although background and specimen are appearing bright in this case, right? So there is something called phase contrast microscopy. This is used, let's say, whenever the light passes through the sample, because there are two kinds of changes that happen. One is there will be magnitude change, and the other one will be contrast change. So the phase contrast microscopy, what it does is it adjusts the contrast so that the images are seen very clearly, so it can avoid any blurry images, right? So for that purpose, this phase contrast microscopy is very much useful. There is something called fluorescent microscopy. This is very much in use whenever you work with the stained samples, a sample that contains dye or fluorescently labeled samples, right? In those scenarios, fluorescent microscopy is very useful. Confocal is useful whenever you want to obtain high resolution images, by eliminating out of focus light. You can also construct 3D image, right, of a sample using the confocal microscopy. So this dark field microscopy we have already seen. The difference here is, because it collects only the scattered light from the sample. Okay, you are able to block the background light coming from the sample. So okay, in this way you are able to see that only the specimens, that is the samples, or whatever light that is scattered from the sample is only collected. The rest of the information is blocked in this case. That's why the specimen objects bright, uh, you know uh, uh, you know uh, whereas the background appears dark in this case, right? Right. So the fluorescent microscopy that we see, that we talk about, is basically it has what it has magnifying power lens, right? And a light source, that is basically mercury or xenon lamp. Unlike the bright field microscopy, you will have to use mercury or xenon light source. This will be useful to excite the fluorescent part, right? So whenever this fluorescently labeled molecule or the object absorbs the light, it will emit the light at a different wavelength, right? At a longer wavelength, okay, right? And this will be collected by the specific filter, okay, that are available in the microscopy. Okay, let's say if you want to understand a sample, okay, stained sample, you can use a specific filter, okay, so that except those light of that particular wavelength only will be allowed to pass through. The rest of the wavelength will be blocked. So in such case, you will only be collecting the information about that stained specimen. okay, or fluorescently labeled specimen. This is very useful, to visualize a particular a part of the specimen sometime, okay, are only those which is fluorescently labeled, okay, right. Yes, so here comes the electron microscopy. Why electron microscopy one can use, right? So the answer is when you want to resolve a specimen or object, okay, up to zero point 0.1 nanometer, okay, uh, electron microscopy is quite often used, right? So we have just now seen that light microscopy has some limitation that comes from the wavelength of light itself. Okay, so we have seen that based on the Abbe's diffraction limit, even if you set the wavelength of light as 500 nanometer, what you will be getting is the any objects which are kept at a distance of, let's say, 178 nanometer is what can be resolved using the light microscopy. Okay, whereas in the case of electron microscopy, one can resolve samples, any objects which are kept at a distance of 0.2 nanometer also. Okay, so this resolution comes from the magnifying power of the electron microscopy, that is nothing but 20 lakh times, right? You can use the electron microscopy to magnify up to 20 lakh times, whereas the light microscopy offers only up to 1000 times or 1500 times. That is the difference. So why this difference occur is because when electron beams are passed on to the sample, electron can access anywhere between the atoms. Okay, so the characteristic size of electron, or the de Broglie wavelength for electron, you can just calculate based on this high voltage, that is 200 kilovolt, the lambda for electron in this case will be 0.0025 nanometer. So that is the characteristic size of electron. So electron can access any space between the atoms, whereas light cannot pass through because of the wavelength itself, right? Because of its limitation, right? So that is the difference. So when it comes to electron microscopy, yeah, let's start with the SEM, scanning electron microscopy. It has got electron gun, okay, and through electron gun, the beam of electrons are generated, and that pass on to the sample. As electrons interact with the sample, it produces two kinds of signal. One is backscattered electrons, the other one is secondary scattered electrons. There are two detectors. These two detectors collect the backscattered electron and secondary electron. Both these signals are collected and processed to form an image that look like a 3D image, even though it is a 2D image, right? So what is transmission electron microscopy? How it is different from the scanning electron microscopy? The only difference is the beam of electron transmits through the thin sample. Okay, as it traverses from the sample through the sample, it not only collects the backscattered electrons and the secondary electrons. In addition to that, there are something like elastically scattered as well as inelastically scattered electrons. The elastically scattered electron meaning after interacting, they interact with the atom, okay, and nuclei and other aspects of the sample without losing energy, whereas inelastically scattered electrons interact with the sample but it loses the energy, right? So these two electrons, in addition to the backscattered and scattered electrons, help us decode the information from the sample, that you can collect you can get the information about sample structure, crystal orientation and atomic arrangement, right? In addition to the features that we have uh, in the case of scanning electron microscopy, with the help of transmission electron microscopy, you can decode this many information as well from the sample. That is the advantage of transmission electron microscopy. Here we will look at the scanning probe microscope. Usually it involves scanning a sharp probe over a surface to gather information about the topography, okay, properties and behavior of materials at very high resolution and at the atomic level. So there are several types of probe microscope. Okay, widely used are scanning tunneling microscope and atomic force microscope. Other probe microscopes used are listed over here. Okay, how scanning tunneling microscope works? So often it is called as STM. Okay, and we also call atomic force microscope as AFM. Now, how scanning tunneling microscope works? Usually there is a very sharp tip, okay, attached to the cantilever, okay, and when you bring this tip very close to the surface, maybe at a distance of, let's say, 1 nanometer, okay, there is something called quantum tunneling happens. So which means that there will be a flow of electron from the tip to surface or vice versa. Now as you move along the surface while scanning, whenever there is a change in the distance occur, okay, and that will be reflected in the flow of electron. So this change in the electrons signal collected by the detector is again processed to form an image. Okay, that's how the STM works. The advantage of STM is you can actually collect high resolution three-dimensional topographic image. Okay, but the disadvantage part is you need the sample need to be conductive. I mean conductive, right? Sample must be conductive. So that is the one disadvantage. Apart from that, the atomic force microscope, okay, can also be used, to collect the surface topography and surface morphology, right? So it works in a three different modes. One is contact mode, non-contact mode, and tapping mode. Okay. So how it works in contact mode? Uh, so how it works is there is very sharp tip attached to the cantilever. So here the tip will actually touches the surface. Okay. So when it touches the surface, in three-way force will be experienced by the tip. One is due to contact force, or van der Waals or electrostatic force due to interaction with the atom and molecule at the surface on the surface. Now when this force is experienced by the tip, this force will cause the cantilever to deflect. Okay. Now there is something called laser light that will be sent at the back of the cantilever. Now this deflection is traced by the laser light, right? As and when you scan the surface, whenever there is a change in the force, that will cause changes in the deflection as well. Now this deflection information is collected by the photodetector, okay, and finally this information whatever gathered by the photodetector is processed to form an image. This is one way. Here the radius of the tip that is usually employed is between 5 to 20 nanometer, whereas the tip, the material construction, I mean the material of construction of the tip is usually a diamond, tungsten, gold and platinum. Okay. In the case of non-contact mode, it works because the tip hover above the surface with a distance of, let's say, few nanometer. Whenever there is a change in the distance occur, okay, the change in the amplitude and frequency is collected by the photodetector, this information is again processed to form an image. Okay. In the case of tapping mode, uh, the tip doesn't touch the surface continuously, but intermittently, right? So it will tap at a regular interval. At the same time, the tip will hover above the surface with a distance of 2 nanometer. In this way, you collect the information again and process to form an image. Okay. We will stop here. We will continue from the next lecture. Thank you.